First of all, welcome everyone. Thanks for joining. So for anyone who might be attending this call for the first time, we hold this industry-wide call each month with companies who essentially want to contribute to the cultivated meat value chain. The purpose of these calls is to offer new and existing cultivated meat stakeholders an opportunity to learn from each other, especially with respect to obstacles that they may be encountering and potential solutions that might exist along the value chain. Hopefully with the end goal of really helping to accelerate R&D and commercialization in the field. As noted in my email earlier this week, at the advice of our legal team, we've made some slight modifications to just the advice that we give to the presenters on this call. So this means that the presenters really should aim to structure their talks kind of as if they were giving a presentation at a scientific conference, and to avoid coming across as just, a little too salesy. And so we hope that this adjustment won't be very difficult to implement and that the seminars will still sort of remain equally valuable to you. And we'll let you know if there's any other changes moving forward. If your group is interested in presenting on a future call, or maybe you're working with a company that could benefit from being on these calls, do feel free to put them in touch with me directly, and I'm happy to sort of get them added. So before we begin, just a few housekeeping items from our side. I want to first turn it over to Maddie from our policy team for a few updates.
Thanks. I just have one update. I want to share that GFI has a new webpage and newsletter that provide information on U.S. government funding opportunities for alt proteins and startups. So I'll drop a link to the webpage in the chat, and on that page, you can both explore current funding opportunities and also sign up for a newsletter so that your company doesn't miss out on future sources of public funding. I'll hand it back to you, Elliot.
All right, thanks, Maddie. And I also wanted to share sort of along those same lines, our corporate engagement team recently released a new guide on financing, as well as a sort of guide for helping decision-making related to scale up and commercialization. So that's a new report that we have out that might be of interest to folks that are, at that point in which they're making decisions around scale up, selecting a CDMO, how to finance the facilities that you might be planning, et cetera. So please feel free to download the report at that link there that I put in the chat. Alrighty, so without further ado, today I'm pleased to welcome Kiriako Tsoukalas from Hydrosome Labs and his colleague, who will share findings outlining how nanobubble technology could mitigate maybe some of the challenges related to scale up in your bioprocessing. So as always, feel free to drop questions in the chat throughout, and we can get to them at the end of the presentation. So I will turn it over to you.
Cool. Thank you, Elliot. Yep, my name is Kiriako Tsoukalas. I'm a scientist and one of the directors of BizDev here at Hydrosome, and I'm joined by Nick Jackowetz. He's, like I said, our North Star, our CSO. Nick, you want to do a...
Yeah, pleasure to be here, everybody. Nick Jackowetz, Chief Scientific Officer here at Hydrosome. Really excited to talk to you about our ultrafine bubble technology today and get into this really exciting and emerging technology. So excited for the presentation and the questions to come. So with that, I'll hand it back to Kiriako.
Cool. So we're obviously in the same room. So for the first part of the presentation, I'm going to kind of shift the screen over to my end. And then if I can share my screen. So I think just to start off the call, I mean, it's evident that biomanufacturing, cultivated meat, fermentation, alternative proteins is the future. That's why we're all in this room, because of our innate belief in the space. And with that being said, I mean, there's some real key cost drivers in these processes, in biomanufacturing processes, regardless of what industry or what process that you're focused on. So raw materials, I mean, the cost of substrates, of nutrients to use are quite expensive. Energy consumption throughout, all the way from the upstream, all the way to the downstream, electrical requirements, agitation, heating, cooling is also a cost driver. And then labor too. Again, all the way from the upstream microbiologist to the fermentation engineers, to the bioprocess engineers, all the way down to the downstream scientists. They are not cheap, and rightfully so. Typically, I open this up for a discussion, but hopefully this is something that resonates with all of you. And ultimately what this leads to, especially in the alternative protein, cultivated meat space, is just high per unit costs that are not competitive or just partially competitive with the market alternatives. And ultimately, to address these cost drivers, the innovation has been focused on really two different areas, right? So upstream development, so strain engineering, different substrate engineering, to really maximize the titer, maximize the robustness of that strain, and increase that product of interest. And then conversely, on the downstream side, I mean, companies are built on their different extraction methods, their purification methods. But regardless of the approach, there are really some key bottlenecks that I think GFI has done a great job in highlighting too with their reports. I mean, insufficient capacity is globally a huge problem in the space. When you get to the commercial sizes, I mean, poor gas transfer in these processes obviously lead to lower yields, lower titers, and ultimately a lower production of that product of interest and really high operational costs. So there really needs to be a solution that can address these operational bottlenecks and be additive to the innovations, to the R&D, to the efforts in upstream and downstream developments to ultimately achieve that target pricing. And what we've found is that the solution to this is within a portion of the biomanufacturing process that is often overlooked. And that's the growth process. So, you know, when you really look at it, there's really been no significant advancements in additive bioreactor technology for quite some time. I mean, with that being said, companies are well established in commercializing and providing increased capacity for smaller startups. And there are also companies that are focusing on continuous fermentation and different kind of advancements in the space. But in terms of technology that really retrofits into current reactor infrastructure, I mean, there's been new sparging methods, there's very new retrofitting techniques that people have had, but ultimately none of them have quote unquote stuck. They've been met with very little success. And the solution to overcoming these bottlenecks is within that growth process itself. And we believe that the key to that, to enhancing the growth process is really enhancing current tank infrastructure, right? So there is a need for a technology that can easily retrofit into current reactor systems, into current tank infrastructure, a technology that's scalable, that has a low carbon footprint, that can work anywhere from a benchtop all the way up to the commercial scale, and that can ultimately enhance gas and nutrient transfer to cells to unlock these bottlenecks and ultimately increase the speed of these processes and maximize production capability. And up until this point, there really hasn't been a technology that has hit on all these markers. And that's where we believe ultrafine bubble technology really is an answer and a solution that could be quite valuable in the space. So just a few slides on the background of Hydrosome and what we are. So we're a biotech company. We have a patented platform processing technology. So our tech is about the size of a small refrigerator. It is a purely mechanical process. It's a very minimal carbon footprint. There's no chemical additives. And ultimately what we do is we hit solutions. Solutions flow through our technology. They're hit with finely calibrated physical forces. And these forces create high concentrations of ultrafine bubbles. And ultrafine bubbles, we found, enhance nutrient transfer and potentially enhance gas transfer to cells. And within biomanufacturing processes, that's really important. So to get into kind of a brief overview of how our tech really works. So again, it's about the size of a small refrigerator. So solutions enter our technology. So within a biomanufacturing processes, this would be the media, this would be the cells. They would go through our technology. That solution is hit with these finely tuned forces. There's no loss of cell viability that we've seen based off the studies that we've conducted. So the cells are safe through our technology. And ultimately what these forces do is create high concentrations, like I said, of ultrafine bubbles. And when in solution, these bubbles are very effective carriers of nutrients and potentially gases to cells. So just a slide here on really the industries that we're focused on. It's with a platform technology, there are a lot of areas that we could touch. But with that being said, really we've honed it down to two. So consumer products really doesn't pertain to this call, but we are commercializing the cosmetic space right now. So when you use ultrafine bubble water in cosmetic formulations, these bubbles are extremely stable. They have a long shelf life and they increase delivery of active ingredients and hydration. So in clean and natural beauty, that's really important. We're doing a little bit of work on the beverage side for different organoleptic effects and different health benefits. But the reason that we're on the call today and the reason that we're so closely integrated with the GFI ecosystem is fermentation, is biomanufacturing. So we found that when you use, and we've conducted studies using our technology in these processes, and we're showing significant enhancements in production efficiencies. We're talking shorter process times and higher yields. So with that being said, this is the exciting part. I'm going to hand it over to our CSO, Nick Jackowetz, and he's going to get into a little bit of the theory of ultrafine bubbles and go through some of the data and the studies that we've conducted as well.
Great. Thanks so much, Kiriako. Really appreciate it. And again, thanks for everybody for having me today, and pleasure to be here. So yeah, to get more into the science is where it gets kind of fun and really interesting. This idea of ultrafine bubble technology is relatively new in the scheme of science. So it really goes back to the the 80s in Japan, where these bubbles were first looked at and first started to be discovered and measured. They were found in the ocean, and since then there has been quite some significant increases in interest in these ultrafine bubbles, especially in the last 10 to 15 years. So you can see the peer-reviewed publications there, in the 90s and 2000s was pretty quiet, but it's really taken off in the last little while. And that's because our ability to be able to measure these ultrafine bubbles has gotten much better. As you can imagine, these bubbles are very small. So when we talk about how small they are, they're about 100 nanometers or so in size. So to put that in perspective, that's about the size of a virus particle. So very, very tiny. And they are bubbles, so they are voids. They don't have much mass to them. So their ability to be detected by even optical methods is a bit tricky, especially when they get really, really small. And so that's been a hindrance of our ability to be able to measure them and to be able to use them effectively. But now with more advanced tools, more advanced optical tools, and even electron microscopy, we can see these bubbles much better and get a sense of what they are, their concentration, their distribution. And especially only in the last few years, you see even ISO step in and put some framework around what ultrafine bubbles are. And ISO's define that as a bubble that's less than 1 micron or 1,000 nanometers. So you can see there's some coalescence around this field in the last little while and a lot more interest in this technology. Typically, in the past, this technology was used mostly in agriculture, so to help plants grow more effectively, especially in indoor ag, to really helping move nutrients and gases to the root systems of plants and help make them grow with less fertilizer and more quickly, and also in wastewater treatment, where they were used to help with waste streams there, help with flocculation of different waste streams that are there, and also increased oxidation rates in some cases too. So it is a new technology, and at Hydrosome, as Kiriako mentioned, we're focused on bringing this technology to new verticals, the consumer packaged goods space, fermentation, and even life science. So it's a really exciting time in this ultrafine bubble space for us. As I mentioned, we think about a bubble. We typically think about a bubble that is transient, something that we see in our champagne or in our sparkling water, something that is there for a moment and then floats to the surface. Ultrafine bubbles are different. The physics when we get this small change. And so ultrafine bubble, again, at that 100 nanometer scale is unique because it's not governed by buoyancy anymore. It's governed by Brownian motion. So these bubbles that we're talking about, they do not float. They can remain in solution for quite some time. And that's a really interesting and important property of ultrafine bubbles. They also, because we can make many, millions and billions of them per milliliter, they have tremendous surface area. So we have a lot of these bubbles that we can add, and we can vary that concentration of those bubbles and really maximize surface area. So that maximizes the interaction of cells with these bubbles and also chemicals as well, too, if we're thinking about chemical reactivity. The bubbles themselves are formed, as Kiriako mentioned too, through force and pressure. So we're not adding any surfactants or any other chemicals here to this mixture. It's really just through these principles that we make them through hydrodynamic cavitation. And that creates these bubbles. The interface of the gas and the liquid has a layer of hydroxide ions that are naturally present in water, and these form spontaneously during our process. And so that's the boundary. And because they're hydroxide ions, these bubbles do have a slight negative charge, which means they repel each other. And that, again, is something else that helps with their stability. So they don't want to coalesce and Oswald ripen over time. They do actively repel. And along with their decreased buoyancy is a hallmark of their stability over time. Another interesting property of these bubbles is the pressure that is inside of them. As we shrink a bubble down, something called the Laplace pressure, due to the radius and the curvature of the bubble, the radius gets very small. And so the internal pressure theoretically can be quite high in these bubbles. So we're talking about a bubble that perhaps is 100 nanometers or so in size. And theoretically, filled with air, that bubble would have a pressure around 30 atmospheres. So their gas carrying capability could theoretically be quite high. And that's very interesting, especially if we think about trying to deliver gases, especially oxygen, to cells that need it. Again, as I mentioned, this technology is quite sustainable. There's no chemical inputs. Even the power used to drive our technology is very minimal. So for example, to process 100 gallons of fluid takes about 2 cents worth of electricity at Illinois rates here where we are. If we think about this technology and what we are doing with it and really trying to push it, it's really interesting, and there's so much potential, I think. There's not many companies doing what we're doing right now. Again, the history of ultrafine bubble technology has been in wastewater treatment and ag, and so we're really pushing it into new areas, but really focusing on innovation as well, thinking about how we can adjust concentrations of these bubbles to get them higher, to have even more surface area, even more interaction, and more, say, gas carrying capability. We're very focused as well on looking at the addition of other types of gases inside of these bubbles. So right now, most of our bubbles are filled with ambient air, so mostly nitrogen and oxygen. But we can produce bubbles with exogenous gases as well, say pure oxygen, and which would be especially interesting for aerobic fermentations, or maybe CO2 for anaerobic fermentations, for cleaning purposes. Also, we've had other types of industries be very interested in this technology when it comes to beverage applications, for example. If you can fill an ultrafine bubble with, say, CO2, it doesn't float. It has tremendous surface area. It's not going to come out of solution. What does that taste like? What does that do to your perception of flavors or mouthfeel? And I think these are really exciting areas that, here at Hydrosome, we're really excited to pursue, and we have a lot of active research in this space. So not just, again, the cosmetic space and fermentation, but so much potential here, which is really exciting. I mentioned the stability being very, very strong. So we've seen these bubbles persist for up to 1 year in aqueous solutions. So in water, they can remain for quite some time. And we do a lot of work in cosmetics. And so we've had topical creams and cosmetic formulations where we've seen these bubbles persist for over 2 years. So again, very, very stable solutions, which again is a hallmark of the technology. We continue also to work with leading researchers in this space looking at how to characterize and measure ultrafine bubbles. So we have collaborations at the University of Illinois, Chicago, and the University of Chicago looking at ways to use electron microscopy to measure ultrafine bubbles. And we do this through cryo-TEM, so freezing the samples, and also something called liquid cell transmission electron microscopy, where we can look at a liquid sample and quantify these bubbles. And through that, we've noticed bubbles as small and even smaller than 50 nanometers, and smaller, some of the smallest that have ever been imaged in peer-reviewed literature. That's collaborations we're working on right now to bring that to publications. And so it's really exciting, and these smaller bubbles may have unique properties and may have just different things and different applications that we were not previously aware of. So it's very exciting time for us. But when we think about this tech, like how does it work? Always, as scientists, we always think about what is the mechanism of action, what's happening here. And I will say that is an ongoing area of research in the broader scientific community of what's happening. What we know today, and I'll walk that through that now, to go through that idea, but it's still developing thoughts. Like we're still working on these exact mechanisms, but we'll walk you through what we know today. And some of that is a concept that we call attract and deliver, where we have many of these bubbles in solution. Again, they're very stable. They have a charge. So there could be, we understand there could be some electrostatic interaction between the particle, the bubble, and other types of solutes in solution. So attracting, say, positively charged ions and species to the surface. We're moving these species and solutes more effectively with these bubbles through that liquid, through that solution, and bringing them in closer proximity to cells where perhaps their bioavailability is increased, or just having those bubbles, say, stick to that cell surface and along with those solutes, so that cell has better access to them. So we're learning more about that delivery of these different types of solutes and gases as well to the cell. And so that's a theory. We've also been looking at the effect of cells and how cells respond when in the presence of ultrafine bubbles. And we've been doing a lot of RNA sequencing. And this gives us a snapshot of what's happening inside of that cell and how they respond. So we can compare, for example, here, a cell like E. coli to a solution where they have ultrafine bubbles and one that does not, and look at what's upregulated and what's downregulated in that cell. Like, what are they doing in response to those bubbles? And we see things like much higher increase in transport mechanisms. So we're doing a lot of research right now looking at how these bubbles can affect transport, but we're seeing, better permeability of lots of different types of solutes. We're seeing upregulation of genes involved in nutrient that are upregulated when there's higher nutrient availability. So it seems like these cells have better access to nutrients and are actively making and biosynthesizing lots of compounds. So we're looking into that as well. We see genes upregulated as well when it comes to gas availability, specifically oxygen. So we see genes around the electron transport chain. We see genes involved in oxidative phosphorylation and even the TCA cycle being upregulated, which may speak to better availability of oxygen, whether that's through the bubble itself or whether that's through other means. We're still investigating those mechanisms, but that's very exciting, because if we're able to drive better oxygen availability, obviously that has very important implications for energy production. And then finally, on that note of energy, we do see a lot of precursors involved in the ability of these cells to produce energy, ATP and other precursors that are upregulated as well. So overall, we're seeing what we call this sort of focused growth state that these bubbles can induce. And it's very interesting, and we're early days when it comes to this kind of interpretation, but it's very exciting. And I'll walk you through some data, very shortly of what we've seen. But I think for us, it's always seeing is believing. And I think we have lots of different tests that we do to look at these bubbles. But one that we do enjoy, and I think articulates this well, is something we call the M&M test. So we definitely enjoy some candies here at Hydrosome and like to use them in different ways. And here's a cool way to kind of look at these ultrafine bubbles in real time. So here we have two solutions: deionized water on the bottom and hydrosome water on top. The only difference here is the presence of bubbles. And we're watching the diffusion of the color off of these M&Ms. And you can see on top, we have better and more efficient and faster diffusion of that colorant off of those M&Ms versus the deionized water at the bottom. All things being equal, temperature, the size of the volume of water here, the size of the beaker. We're just really looking at that sort of attract and deliver theory, that movement, that diffusion enhancement with these ultrafine bubbles. That really helps sort of articulate that there is something happening here, and these bubbles are definitely helping to drive the movement of solutes in solution. So let's talk a bit more about some of the data that we've seen. Now you know a bit more about the theory of ultrafine bubbles, this emerging science, this tremendous potential that we have here with ultrafine bubbles. But let's get into some of the data. As Kiriako mentioned, we're really excited and focused on this growth phase of the fermentation process. We know that there obviously has been tremendous efforts and monies put into looking at upstream and downstream processing, and definitely those are obviously extremely important areas. But again, we think that there's lack of innovation and there's potential on the growth phase during fermentation, and that's where we come in. So we're able to introduce our ultrafine bubbles to the fermenter and really drive, that benefit, drive, increased efficiencies of these fermenters and try and lower operating costs through having fermentations finish faster and through increasing yields overall through, again, this increase with these bubbles, with this mass transfer and higher surface area. The way it works for our technology is pretty simple, really. We like to be agnostic to the type of bioreactors that are out there. So we have a piece of technology. Again, it's about the size of a household refrigerator. It varies depending on the tank size, but for purposes today, that's about the size where it is. We can take that next to a tank. We can hook it up, and essentially we're doing a pump over technique. And so we need two ports. We can pull out of one port, bring that liquid, so that broth, the cells included, through our tech. That our technology infuses those ultrafine bubbles and then reintroduces that stream back into the tank. And we do this repeatedly. And this is beneficial because by repeatedly adding and putting the cells in the broth through our technology, we can keep adding more and more ultrafine bubbles. So this process is additive. So as the fermentation continues, as bubbles are perhaps used up or consumed, we're able to keep making more. So we're keeping that surface area up and actually raising that bubble level even higher. So that's a really really advantageous, and we've seen a dose response on our end, when more bubbles really helping to drive faster and more efficient fermentations. And again, it's a really easy setup. It's sort of a plug and play, if you will. Our technology is automated. It sits right next to a tank, monitors everything. We just need those couple ports and everything is ready to go. Early days, though, we were looking at our technology and looking at batch processes. So looking at just taking some of this ultrafine bubble water, infusing it into a growth media, autoclaving it, and then inoculating it, and seeing what was happening with different cell lines. And we started with yeast and some partners looking at Pichia pastoris and looking at biomass production there. And we typically always see this enhancement of biomass growth with ultrafine bubbles being present. So with Pichia pastoris, we noticed modest 18 or so % increases in that cell biomass over time. And same with Saccharomyces cerevisiae, we noticed shorter lag phases and a faster exponential phase. Even despite some stressors that we added to solution like sorbic acid, where hydrosome technology and ultrafine bubbles would help these cells persist and grow through that. So these were early studies that kind of got us thinking more about fermentation, and more that this was something we needed to look at more seriously, and how do we adapt this really exciting and emerging technology to the fermentation field. We've also been working with lots of different types of cell lines. So even filamentous fungi with folks doing alternative proteins and looking at producing this filamentous fungi as the biomass itself. That's the end product. So where we did some work with a group there and saw benefits there, significant benefits when it came to the growth of that biomass, just simply having those ultrafine bubbles present. So we think there's tremendous area to innovate and increase efficiencies and growth here as we kind of dial in our ultrafine bubble technology to these different systems. And again, these are batch processes. So we're just adding these bubbles in at the start. We're not continuously adding them. And so I think there's real potential to really further this by infusing these bubbles constantly. And that's where we started working at the pilot scale with the University of Illinois IBRL group. They have a fantastic facility down in Urbana-Champaign. And what we wanted to do is bring this to scale and one, look at infusing ultrafine bubbles continuously during the fermentation. And then two, have it at scale. So have it at a 400-liter pilot scale that is more relevant to commercial applications. And the idea here was to take E. coli cells, BL21 strains, and look at how they are affected by ultrafine bubbles, and look at their growth characteristics, viability, overall fermentation time, and energy inputs, for this process. And so this looked like this. Again, we were taking that tank, we were setting up our technology right next to it to infuse ultrafine bubbles continuously, and then looking at that growth. Conversely, for the control, we had a setup very similar, where we did have a pump over, we just bypassed our tech. So we were still mimicking the pumping action, but we just weren't having all of the cells and broth go through our technology, and so thus no ultrafine bubbles. And we wanted to look at the end product of what was happening in those growth curves. What we noticed was differences in overall growth in the growth rate. So with the hydrosome situation, the hydrosome treatment, the light blue line shown there, we see a shorter lag phase. So that fermentation has really started to hit exponential phase around 30 hours and take off there, versus the control really only started to take off around the 40-hour mark and then move from there. When these fermentations were concluded as well, we looked at the cell count, or the amount of cells produced, and we saw about a 2x increase in the amount of cells that were there when we compared the hydrosome treatment having twice as many cells versus the control. And so this really got us excited and started further collaborations looking into, again, a fed batch fermentation, a larger size, that got us looking further into how we can affect other types of cells, and just that these benefits are real and definitely are real at this scale. When we looked at the doubling time as well with this type of fermentation, we noticed that the doubling time during exponential phase was around 50 minutes in the hydrosome case and about 100 minutes in the control. And so we saw, again, a twice as fast doubling in that situation. We also looked at what we call the glucose utilization rate and efficiency. And we saw, again, that the hydrosome treatment was able to take glucose and more effectively convert that into biomass versus the control by about a 3-fold difference. We also then took that information, as we alluded to before, and really tried to understand what's happening on that molecular level. And so we did some RNA sequencing, pulling those cells from both of these treatments. And again, looking at that upregulation and differences between the groups. And so here's kind of just a quick, high level snapshot of what we saw and the percentage of genes that were changed, but a lot around nutrient availability, genes that are involved and switched on when there is more of abundance of nutrients that are available to these cells. And that was very clear here. And it's same with biosynthesis pathways, making more things like amino acids and proteins were upregulated, things around energy management and optimization, membrane transport, again, bringing things into the cells more effectively, and then and then genes around oxygen availability too. So there's a lot here, and still lots that we're researching and doing some publication work at the moment on to bring this to light. But some fascinating results here that we're continuing to work on and to do this work in other cell lines as well. So there's much more to come, and we're really at the cusp here of some exciting discoveries and mechanisms that are coming. So to sort of wrap this up too and think about what we saw. Again, we're thinking about efficiency gains here, the ability to use these ultrafine bubbles to really decrease the overall time of fermentation, so speed up how fast these cells are growing and how fast they're moving. Looking at increasing the overall peak biomass that's possible here by making a more efficient route of fermentation. But really thinking about using this technology as a simple add-on to what you already have. So using your current infrastructure, infusing these ultrafine bubbles so there's no added chemicals here, just thinking about these bubbles and added surface area to really bring these efficiencies and some surprising notes as well. Again, thinking about the movement of nutrients more effectively to these cells with the bubbles, but also there seems to be a recognition of the bubble by these cells, and there's definitely a response that we've seen in a few cell lines here. So it's not purely as simple as just moving nutrients. There's definitely a cell response that mediates some other types of metabolism shifts, and so we're really excited to keep looking into that. And so it's a very exciting space for us right now, and we're really looking forward to further research. And this is where we do a lot of work with collaborators to bring this technology to their process and see how it can be additive to them. And so again, with that, I think we're really, again, excited about this technology, about what it unlocks in terms of the overall fermentation process, the ability to really look at that growth phase more closely and help, increase optimizations there, enhancing, that gas transfer, mass transfer overall to help, really speed that growth and help increase titers and yields at the end of the day. So it's a very exciting tech, and we're really, really pleased to be able to talk to you guys about it today and again, collaborate with different researchers and groups that are interested in using this technology with their own process. So I think with that, maybe I just say obviously thank you very much for everybody's attention today. And for Kiriako and myself, I think we're again super excited to answer any questions that you may have. I think Kiriako just wanted to mention maybe one thing before we end.
Yeah, thank you, Nick. So on that study that we conducted with the University of Illinois, if any of you are interested in reading kind of a little white paper that we put together about it, I'm going to drop it in the chat once we exit out from the screen. But again, yeah, like Nick said, thank you all for your time, and we'd be happy to answer any questions you have, and then maybe just put our contact information, emails, if anybody wants to reach out, has any other questions or wants to start a discussion. Excellent.
Yeah, thank you so much, Nick and Kiriako, for that presentation. And indeed, I think some of the principles that you discussed, I'd be very curious to see, how they might apply to mammalian cell cultures, etc. Alrighty, well, that was a great discussion. I think we're at the end of the hour here, so yeah, thank you again for presenting. I'd love to see folks, try to replicate some of the data that you have for yeast and bacteria in animal cells. I think that will be really interesting. It seems like the principles might be conserved there, but the experiments will bear that out, hopefully. So yeah, thanks everyone for attending and contributing to the discussion. And yeah, we'll see everyone here next month.
Cool.
Thank you.
Thank you.
Appreciate it.
Thank you.
Bye.